Hi everyone, good evening guys. Welcome back to Unacademy NEET English channel and my name is Tina. I'm your biology educator. So guys, yes, Re-NEET is finally of course happening and we have already started our preparation for Re-NEET in Bounce Back series. Also, you must know that we have also started the All India mock test series, right? So earlier yesterday I told you that these mock tests are happening every like twice a week, every Wednesday and Sunday. But now these days have been increased, guys. These days have been increased so that we can prepare better. Yes, please. So that we can prepare better. And in order to make you practice more, so guys, now these mock test series will be occurring thrice a week. So every Sunday, Wednesday, and Friday. Timings would be exactly same as that of NEET, that is 2 p.m. to 5 p.m. So don't forget to enroll for it. So yes, guys, let's get back to our bounce back series, and we are talking about zoology of class 12. So we have started with part 1 yesterday, and in part 1, guys, we have started with the human reproduction chapter. Good evening, all of you. Quickly, let me know how many of you have attended that with me. Yes, guys. How many of you have attended this? Good evening, Ji. Good evening. Yes, yes, yes. You have link to enroll. So link to enroll is given in the description box, guys. You can easily enroll for the All India mock test series, and they are very, very important because they not only help you practice, but practice under pressure. And also you will get an All India rank. So it gives you a rank prediction also, where you really stand. And also you'll be able to analyze because video solutions will be available for every test. So all these things are important. Just mere studying in blankness doesn't work, guys. So you have to practice mock tests for before going for the real NEET exam. Good evening, good evening. Hi Mohan SR, so lovely to have you in the class. All right, guys. All right. So yes, guys, we have started with human reproduction. So I'll tell you what we have done in the previous class. So we have discussed all the details, guys. We have discussed all the details of the structure of male and female reproductive system. We have also studied the process of sperm production and ovum production, that is oogenesis, and how the two processes are different from each other. We have discussed it all. See, and also the hormonal regulation in both males and females have been studied, right? So this was the female structure. We have discussed that in detail as well, and I have discussed with you the process of oogenesis also, right? So yes, this wait a second. Yeah, this diagram was also very, very important, where we have seen the structure of primordial follicle, primary follicle, secondary follicle, and how every follicle is different from the other follicle, right? So we have also seen yesterday that these till here, sorry. So guys, till here, the process happens during the fetal stage. After fetal stage, that is after our birth, no new follicles are added. This is a very important line. And hereafter, so this is till birth. So this is before birth or till birth. Wait, this is before birth. And while we are born with around how many follicles? Primary follicles, 1 million follicles in each ovary. And at puberty, out of that 1 million, only 60,000 to 80,000 primary follicles are left behind. So we have discussed it all, guys. Please tell me that we are thorough till these points. We are thorough, right? And now the journey of the primary follicle becoming secondary follicle and tertiary and then the Graafian and so on. This process occurs every month. Every month, one follicle out of the 60,000 or 80,000 develops further, and this process becomes or is known as menstrual cycle. So in menstrual cycle, guys, we have seen there are so many implications. In menstrual cycle, there are events occurring at different levels, So when we study the menstrual cycle, we've seen that there are events occurring at the uterine, within the uterus, so we call them uterine. Then ovarian hormones, how ovarian hormones fluctuate, what are the events occurring within the ovary, and how do these pituitary hormones, that is FSH and LH, fluctuate during the entire cycle. All right, so let's have few conclusions here. Let's draw some conclusions here. So basically every menstrual cycle begins with the, so in the beginning of every menstrual cycle, guys, beginning of menstrual cycle, I'll write an MC, right? Menstrual cycle. Now see, the level of progesterone falls. So as the progesterone falls, FSH will be high. Progesterone is very low and FSH is high. It is FSH, high FSH, which is responsible for the beginning of cycle. Because during the high FSH level, a new follicle will be stimulated to complete its journey, right? And during the end, again, the end of menstrual cycle is marked by the beginning of the next cycle. Is that clear? Then ovulation is majorly because of LH surge. It is because of a sudden increase in LH, that is called LH surge, right? The hormones which show two peaks, so hormone which shows two peaks, that is estrogen, guys. That is estrogen. This is the only one hormone which will increase to its maximum twice during the cycle. And these two peaks, see, these two peaks, one will be in pre-ovulatory phase, that is during the proliferative or the follicular phase around 11th and 12th day, while the other peak will be in the post-ovulatory phase, which is also known as the luteal phase, right? These, and out of these two also, maximum increases during the pre-ovulatory phase, and it is this high estrogen which results in or which causes LH surge. Please tell me all these points are clear. So make sure that you write down all these conclusions in your NCERT or in your notes. Is that clear? So guys, we have discussed this in detail, and we've also talked about these. The first menstruation is known as menarche, and it begins at puberty, which arrives around 11 to 12 years of age, right? Now, we have talked about, see, this is the new topic that we are discussing. So, sperm production has been done, and also oogenesis is complete, and we know that not complete. So we have studied oogenesis completely, but we have also read this point that before sperm entry, see, let's come back to this topic. Wait, we have discussed these points. What is happening here? Yeah, on to this. So see, we have seen that during ovulation, it is not the ovum that is released. During ovulation, it is the secondary oocyte and the first polar body that is released. Yes, it is released. Is that clear? Now, secondary oocyte is going to enter into the fallopian tubes, right? And now fallopian tubes is that site, particularly it is the ampulla of the fallopian tubes where the sperm must reach for it to fuse with the secondary oocyte. So the fusion has to occur between the secondary oocyte and the sperm, and it is after the sperm entry, guys, it is after the sperm entry that secondary oocyte will complete meiosis 2 and become ovum. So we have already discussed that if sperm doesn't enter, ovum is never formed. Ovum is never formed. So in today's session, guys, we are going to discuss the details of what happens at the time of fertilization. Let's see here. Okay, now moving on to fertilization. So fertilization, guys, let's see here. So during copulation, semen is released by the penis into the vagina, and this is known as insemination. And we have already read this is the main function of penis. It is a copulatory organ, helps in insemination. Now the motile sperms swim rapidly through the cervix and uterus and reach the junction of isthmus, and it is not exactly, now this is old NCERT, guys, this is not the junction where it occurs at ampulla. It occurs at ampulla, not at the junction. Even if you see some questions in your PYQs, answers might be still ampullary junction, isthmic ampullary junction, but no, it occurs at ampulla. Is that clear? Now ovum released from the ovary also transported to the ampulla, where the fertilization takes place. So where does fertilization occur? In the ampulla region. Is this clear? So again, it reaches the ampulla. Now see here. Now this is a very important point to be noticed. So fertilization will only occur. So this is a PYQ line. Fertilization will only occur when both sperm and ovum reach the ampulla simultaneously. And the process of fusion of sperm and ovum, actually it is not ovum, it is secondary oocyte, which is known as fertilization. Is that clear? Now how does fertilization occur? What are the main steps? Because during fertilization, guys, three things have to happen. Sperm has to cross the barriers around the ovum, right? Then sperm has to penetrate, and after penetration it has to make sure that no other sperm enters, that secondary oocyte has to make sure that, and then the two nuclei of the sperm and the ovum, they have to fuse together. So see, the motile sperm swim and enter and finally reach the ampullary region where it takes place. So this is the location where fertilization is going to occur. Is that clear? Now this is the structure of the mature ovum. So you have to take notice of what are the layers around the ovum or the oocyte. So see, first of all, there is this zona pellucida. There is zona pellucida, and we have already seen that zona pellucida is secreted by, guys, zona pellucida was secreted by secondary oocyte. This is a glycoproteinaceous membrane made up of glycoproteins, and it is secreted by secondary oocyte. Is that clear? Hence it is also a primary membrane. Clear? Then there are some granulosa cells which came along with the oocyte at the time of ovulation, and these cells are known as corona radiata. These are derived from granulosa cells. Is that clear? These are derived from granulosa cells. So outside the zona pellucida we have corona radiata, and then in between the corona radiata, guys, we have this called hyaluronic acid. It is a polysaccharide mucus-like consistency, and it helps in holds the cells of granulosa cells. Cells of corona radiata. Is that clear? Are you getting that? Now see, so that means, wait a second. So let's draw the structure once again. So this is our ovum. Okay, this is the ovum. Watch very carefully, or secondary oocyte actually to be precise. And then we have this layer called zona pellucida. This is the zona pellucida. Is that clear? And in between the two, we have this space which is known as perivitelline space. This space is known as perivitelline space. Is that clear? And then outside this, we have these cells all around it. These cells are corona radiata cells. These are all corona radiata cells, right? Corona radiata cells. Clear? And in between the corona radiata cells, guys, we have this polysaccharide known as hyaluronic acid. Now, when we have understood all the layers around the secondary oocyte, please watch carefully. Now, when we have understood that these are the layers around the secondary oocyte, this is the secondary oocyte. Told you before sperm entry, it will not be known as ovum. Sperm will enter and can transform it into ovum. How, why, we'll talk about it. But for now, this is a secondary oocyte, right? Now, there are so many sperms who are trying to gain entry. There are so many sperms trying to gain entry, right? There are so many sperms. Now, see, for a typical sperm has to cross these three barriers. Yes or no? So, typical sperm has to cross these barriers, which is hyaluronic acid, number 1. Then it has to disperse the corona radiata cells. After corona radiata, it will come in contact with the zona pellucida layer. And after it has come in contact with the zona pellucida layer, right, then it will pass through the perivitelline and touch the plasma membrane. This is the plasma membrane of secondary oocyte. Right? So, this will be the 4th layer that it will come in contact with, right? Now, if the sperm has to cross all these barriers, so it must release certain enzymes, right? So, here comes the role of sperm license. Do we remember who was secreting these sperm license? Yes, please. Do we remember who secreted these sperm license? These sperm license, guys, were secreted by acrosome. They were secreted by acrosome. So, because they were secreted by acrosome, therefore, this is known as acrosomal reaction. So, the first thing is acrosomal reaction. Is that clear? So, first thing that is going to happen for fertilization is acrosomal reaction, where sperm license will be helping in, they help in penetration of sperm into the secondary oocyte. Is that clear? So, these sperm license are of different types, 3 different types. NCERT has not mentioned, otherwise there are 3 different sperm license: hyaluronidase, then corona penetrating enzyme, zona license. All these 3 different sperm license, they will help the sperm to dissolve and disperse the cells and enter into the perivitelline space. Yes or no? Is that clear? Now, imagine this sperm has managed to enter in the perivitelline space and it has come in contact with the plasma membrane. So, as soon as the sperm comes in contact with the plasma membrane, so this is the winner. So, this is our winner sperm. So, whosoever, see, there are so many sperms who are trying to penetrate. All those sperms are releasing the sperm license, but only one will be able to hit this finishing line. So, what is the finishing line? Finishing line is the plasma membrane of secondary oocyte. Whosoever touches the plasma membrane first is the winner sperm. Yes or no? Right? Now, guys, this winner sperm, as soon as touches or comes in contact with, touches the plasma membrane of secondary oocyte, right? Now there will be, as soon as it touches, so touch of the sperm on the plasma membrane will act as a stimulus, and this whole membrane will be depolarized, just like we read about depolarization in nerve impulse, right? This whole membrane will be depolarized. So this causes depolarization of the plasma membrane. Is that clear? Causes the depolarization of plasma membrane. Now the sperm will continue to enter, the sperm will finally enter into the secondary oocyte. That is done. But this depolarization has done one more thing. Now please see, just below the plasma membrane, guys, just below the plasma membrane, we have some special granules in the secondary oocyte. Just below the plasma membrane, there are special granules, and these granules, guys, are known as cortical granules. These are known as cortical granules. So what happens, as soon as the plasma membrane gets depolarized, these cortical granules, they release, they fuse with the plasma membrane, and they fuse with the plasma membrane and fuse with plasma membrane and release calcium carbonate in the perivitelline space. In the perivitelline space. Is that clear? So what happens? All of them will fuse with the plasma membrane, and they are going to release this calcium carbonate. This is calcium carbonate in the perivitelline space. This is calcium carbonate. And from here, this calcium carbonate will be deposited in zona pellucida. And pellucida will become very hard. It becomes, zona pellucida becomes very hard. Now see, while the sperms are still trying to gain entry, they have enzymes for hyaluronic acid, corona radiata and zona pellucida. But because zona pellucida now contains calcium carbonate, sperms do not have any enzyme to dissolve the calcium carbonate. And now guys, after the deposition of calcium carbonate, this zona pellucida will not be digested or dissolved by any sperm. So basically, after the sperm entry, after one sperm has entered, what is happening? Rest of the sperms are banned from entering further into the same secondary oocyte, right? So here we are going to prevent polyspermy. So more sperms are prevented from gaining entry. Now this is happening because of the effect of cortical granules. Hence this is called cortical reaction. This is called cortical reaction. So this is part 2 of fertilization. This is part 1. This is first thing that happens. This is the second thing that happens after the sperm entry. Are we clear? So what is the purpose of cortical reaction? The purpose is to prevent polyspermy. Is that clear? Purpose is to prevent polyspermy. Please tell me these points are clear to everyone. Clear? Now after the more sperms have been banned from entering into the same secondary oocyte. Now see what's happening inside. So the sperm has already gained entry. Sperm entry has happened. Enters. Now what has sperm brought along? So basically guys, sperm has brought along with it the nucleus. This is the sperm nucleus, and it has brought with it the proximal centriole, and it has brought along with it APC. So these are the three things that sperm has brought along with itself. This is the nucleus. We call it male pronucleus, right? This is the primary or the proximal centriole. Okay. I told you the function of proximal centriole will be after the fertilization, and this is anaphase promoting complex. These are the three things that come along the sperm entry. Are we clear till here? Please let me know. Yes guys, are we clear? Anyone with any doubt? Please tell me. Right? Okay. So here the secondary oocyte. So let's talk about the secondary oocyte also. So what does secondary oocyte have? So this is the secondary oocyte. Now secondary oocyte has been arrested in metaphase 2 stage. Told you yesterday that before sperm entry, secondary oocyte is arrested in metaphase 2 stage. So when this APC comes, this is anaphase promoting complex, it will reinitiate the meiosis 2. It will help the secondary oocyte in completing meiosis 2. Help secondary oocyte in completing meiosis 2. Please tell me these points are clear. Right? Now, after completing meiosis 2, now it is no more a secondary oocyte. Now, after sperm entry, guys, this will become ovum. This has become ovum. So this is the pronuclei of ovum now. This is the female pronuclei, that is nucleus of ovum. Are we clear? So what is going to happen here? These two, this pronucleus and the male pronucleus, they are actually going to fuse. They are going to fuse. So guys, this fusion is known as, when the two genetic material will fuse with each other, this is known as amphimixis. This is known as amphimixis. Is that clear? So here the mixing of the two genetic material will take place. Fusion of two genetic material. That is one of father, that is the sperm through sperm, and other of the ova. The fusion of two genetic material. So amphimixis is the third part of fertilization. So fertilization is going to occur in 3 steps. Please see here. First, the sperm is trying to gain entry, penetration, through the help of sperm license, which were produced by acrosome, and hence we call this part as acrosomal reaction. Once the sperm has gained entry, we have to prevent polyspermy. That is, we have to prevent other sperms from gaining entry, and this occurs because of cortical granules by the release of calcium carbonate, and we call this part as cortical reaction. And then, while the sperm has already gained entry, the nucleus, male nucleus is lying here. It has also brought along anaphase promoting complex, which will help secondary oocyte to complete the meiosis 2. After meiosis 2, this will become ovum. And now the pronucleus of ovum and pronucleus of sperm, they will fuse together, and this fusion is called amphimixis. Amphi means two. Mixes means mixing of the two genetic materials, sperm and ovum. This will actually give birth to zygote. So fertilization, guys, is going to produce, this fertilization produces zygote. So zygote is a 2N cell which is produced after amphimixis. Please tell me these points are clear. Right? So guys, this is our zygote, right? And this is the nucleus of zygote now. But we still have our zona pellucida intact. Zona pellucida is still present, and the granulosa cells are also there. Is that clear? So these are granulosa cells, and in between the granulosa cells we have hyaluronic acid. These are granulosa cells. This is zona pellucida. Is this clear? Rest, everything remains same, but now this is no more an ovum or a secondary oocyte. This has become a zygote. Please let me know these points are super clear. Is that clear? And this is the first diploid cell. This is the first diploid cell which has been formed after fertilization. Now zygote, guys, is that cell which will give rise to the whole new organism. Hashtag, it is a totipotent cell. Totipotency means property of a cell to give rise to the whole new organism, and it is the first cell of life, of new life. Are we clear? Now where is it formed? It is present in the fallopian tube. Or oviducts. Now we know that fallopian tubes have ciliated columnar epithelium. The cilia are going to push the zygote towards the uterus, right? Now after around 24 hours, guys, this zygote is going to form. So this is done already, we have discussed this. Yes, so zygote is going to undergo division. See here, so zygote is going to undergo division. This is zygote, the 2n cell, this is a plasma membrane, and we have, wait, let me copy it from here. Okay, wait, this, something is gadbad. All right, let's make that again only. Okay, so this is zygote and this is the zona pellucida, and also has these corona radiata cells, right? So now this is all going to happen in the fallopian tube. Okay, so let's say this is the fallopian tube and here we have our uterus. So this is all going to happen in the fallopian tube. Now fallopian tube is undergoing peristalsis also, there is ciliary motion also, and it is getting pushed towards the uterus. Now after around 24 hours, guys, what will happen? This zygote will undergo first division, and this division is known as cleavage. What is the difference between normal division, normal mitosis and cleavage? We'll discuss. So another 24 hours pass and it will around form first 3 stage and finally 4 cell stage. So this is how the zygote is going on dividing, and from 4 it is going to form these 8 to 16 cell stage. Is that clear? Now while it is dividing, please take notice that zona pellucida is still intact. Zona pellucida is still intact all throughout. Zona pellucida is not going anywhere. But while this is rolling towards the uterus, see these corona radiata cells, since they are loosely bound cells, so during the rolling of this embryo towards the uterus, they are being dispersed. So they are disintegrating, and finally when it has reached this stage, that is 8 to 16 cell stage, right, by this time corona radiata is completely gone. Corona radiata is gone, but zona pellucida is still present. Are we clear? Secondly, so this was the first cell with which life begins, and then this one cell will divide into 2 cells. This is the 2-cell stage. All the stages after zygote formation will be known as embryo. So we are talking about embryonic development here. So where does embryonic development begin? It begins in the fallopian tube. Are we clear? From 2 to it will form 4 cell stage, from 4 it will form 8, and 8 to 16, and so on. Now see here, we call this division as cleavage. This division is known as cleavage. Cleavage is basically mitosis only, because all the cells formed here, they are all clones of zygote, exactly like zygote. But why don't we normally use the term mitosis here? Because you see, with every division, the number of cells is increasing, but the size of each cell is decreasing. Are you getting that? The size is decreasing. Why it is decreasing? Because this is that type of mitosis. Listen very carefully. This is a type of mitosis, guys. Cleavage is a type of mitosis where growth phase is absent. No growth phase. Where growth phase is absent. So that is why the number will go on increasing as usual, but the size of each cell will go on decreasing. And these cells, by the way, are known as blastomeres. All these cells are known as blastomeres. Are we clear till here? And now, particularly this stage, which has 8 to 16 cells, guys, this stage is known as morula. This stage is known as morula. We don't have specific names for these intermediate stages, but we do have a reserved name for 8 to 16 cell stage, which is called morula. Are we clear? Are we clear? Yes. Clear? All right. Now, guys, what will happen the next? See, this is how morula looks like. Now, why have we called it a morula? So, first of all, see, morula means, what does morula mean? When the number of blastomeres is 8 to 16, sorry, here it has to be 8 to 16 cell stage, right? And the characteristic point, it is a solid ball of cells. The cells are known as blastomeres. It is solid ball. That means all the cells are tightly, compactly packed within the sphere as well. There is no hollow cavity in the center. Is that clear? So it is a solid ball of cells. How many cells? It is 8 to 16 in number, and basically morula means mulberry-like. So it is a mulberry-like structure. It is a mulberry-like structure. Now in this morula, guys, in this morula, the zona pellucida is still present. Corona radiata has already been disintegrated. Please be clear about these points. Right? So corona radiata is already disintegrated. Now the next stage is, what will happen? So all these 8 to 16 cells, they are increasing in number. They'll keep on undergoing cleavage. The number of the cells will go on increasing, right? So from 16 it will become 32 to 64. So now next stage after morula what happens? There will be 32 to 64 cell stage. 32 to 64 cells will be formed, cell stage, right? And now this stage is known as blastula. This stage is known as blastula, right? Now what happens in blastula? Basically, see, in blastula, in morula, all the cells are similar. Now out of those cells, there will be certain cells which are smaller in size, right? So the smaller cells, they will be present outside, they will be present on the outer side and form trophoblast. While the larger cells present on inner side will form inner cell mass. Inner cell mass, also known as embryonal mass. Now see here, what's happening? Do we see there are some outer cells shown in yellow? This forms the trophoblast, and the larger cells, guys, larger cells on one corner, they will be arranged. This is known as inner cell mass. Also, do you see that there is a cavity formed in here? So there is this cavity, and this cavity is filled with fluid. So this fluid-filled cavity is formed. Fluid-filled cavity is formed, and this fluid-filled cavity is known as blastocele. Cele means cavity. The cavity of blastula will be known as blastocele, and it is a fluid-filled cavity. Now, because blastula has a fluid-filled cavity, we also like to call it as blastocyst. Now, human blastula is known as blastocyst. See, cyst is a cavity filled with fluid. Whenever we have a cyst in our body, a cyst is formed, it is always fluid filled. So, blastocyst, because it has a fluid filled cavity. Are we clear? Please tell me. Right? So, outer cells, trophoblast, inner cells, inner cell mass, a cavity known as blastocele. Now, trophoblast. What does tropho means? Food. So this is going to provide nourishment. This provides nourishment. This provides nourishment, guys. And what does, this provides nourishment. And what does embryonal mass do? As the name says, why do we call inner cell mass as embryonal mass? Because this group of cells particularly will form embryo. It forms embryo. It forms embryo. So this is another way of showing. So the outer cells, all the outer cells, they represent the trophoblast. These are all trophoblast. On the outer side, this is more like 3D, right? This is a trophoblast. And here we have these cells, inner cell mass, and this is the cavity called blastocoel. Do we understand this? Please tell me these points are clear. Right now, this is the 32 to 64 cell stage. Now this stage, guys, enters the uterus. Very important. Which stage enters the uterus? Hashtag blastula enters the uterus. Please tell me these points are clear. And by the way, the zona pellucida is still intact. So zona pellucida is still intact. Please tell me these points are clear. Now, why are we pressing upon the fact that zona pellucida is present? So, why do we need zona pellucida? So, remember guys, if zona pellucida is removed or it disintegrates anytime before blastula enters the uterus, so the embryo will start sticking to the membrane. It will start sticking to the layers of fallopian tube or uterus. Right? So if this sticking of the embryo happens in the fallopian tube and the embryo enters within the layers of fallopian tube, wall of the fallopian tube, pregnancy will begin there. So remember, this zona pellucida is very, very important, guys. The zona pellucida prevents the embryo from sticking to the fallopian tube. If it will stick to the fallopian tube, it will also enter within it. So it prevents implantation in the fallopian tube. And if the embryo gets implanted in the fallopian tube, pregnancy will begin in the fallopian tube, which will cause rupturing of the fallopian tube. So it prevents implantation of embryo in fallopian tube. Is that clear? So that is why we need zona pellucida. Please let me know that these points are clear. So by chance, if this fails, now see, if this fails to happen, if pregnancy occurs in fallopian tube, it will cause, guys, yes guys, just a second. Yeah, if pregnancy occurs in the fallopian tubes, it is known as ectopic pregnancy. It is known as ectopic pregnancy. So pregnancy anywhere other than fallopian tube, other than uterus, it is known as ectopic pregnancy, and it causes rupturing of fallopian tube, rupture of fallopian tube. Please tell me these points are clear. So if the fallopian tubes gets ruptured, it means one side of the one fallopian tube is gone. So there is 50% chance of the female to get pregnant now, or she is just 50% fertile now. Please tell me these points are clear to everyone. Right? All right, guys. Yes. So that is why we need zona pellucida. Now see here. So we've seen zygote becoming 2-cell, 4-celled, 8 to 16 cell, that is morula, and now gradually it is increasing the number of cells. The size is getting bigger, but the zona pellucida is still intact. And remember, now this zona pellucida contains calcium carbonate. That means it is hard. So as the pressure builds up because of the fluid and because of the increasing number of cells, what will happen? It is putting a lot of pressure on zona pellucida, and this will result in breaking of the zona pellucida. Now let me show you how this is going to happen. Yes. So what happens here? What happens here? So this is our uterus. The uterine lining has become thicker. You know that our body has been preparing for it. The endometrial lining has become very thick during this whole process, and it will keep on getting thicker. So now the zona pellucida will break. Zona pellucida will break, right? And see, the blastula, which has now transformed a bit with outer trophoblast, and then it has also formed inner trophoblast, and the inner cell mass has also changed into two layers, right? And this is the blastocele. See that? This is the blastocele, fluid filled cavity, right? And this is transformation of inner cell mass, upper known as epiblast and the hypoblast. Even if you ignore this, it is fine, but just remember the embryonal mass, inner cell mass, has also increased the number. Now see, there is another cavity which has developed, and this is also a fluid filled cavity. We call this cavity as amniotic cavity and the fluid as amniotic fluid, filled with amniotic fluid. So there's pressure from multiple factors, from blastocele, from amniotic fluid, right? The number of the cells is increasing filled with amniotic fluid. So this causes the rupture of zona pellucida. Zona pellucida breaks, right? And this being the trophoblast. This is the trophoblast. So what will happen when zona pellucida breaks? Trophoblast comes in contact with the uterine wall. So this is all uterine wall, the endometrium, the inner layer known as endometrium. Yes. So trophoblast comes in contact with endometrium and it starts secreting some enzymes. So it releases some hydrolytic enzymes. Who releases it? Trophoblast releases it, right? And this will help this whole embryo to get, it will corrode the endometrial lining, and this whole structure will come to lie within, it will within the endometrial lining. So now this whole structure will embed, it will get implanted in the endometrial lining. So blastula embedded completely in endometrium, right guys? Now when this blastula has completely embedded here, it has completely embedded in the endometrium, guys, this is known as implantation. It has implanted itself. So this is known as implantation. And now see, this is called implantation, and this marks, hashtag, it marks the beginning of pregnancy. Marks the beginning of pregnancy. And when does it occur? From the time of fertilization, implantation will occur within 7 days. So occurs within 7 days or say 1 week of fertilization. Is this point clear? And also this will mark the beginning of pregnancy. So that means after fertilization, pregnancy will begin within 7 days. So now if we test, go for the test of pregnancy, that is that urine test or Prega News we use, right? That is called Gravidex test. So now this will give us positive pregnancy test. Why and how we get that test here? Now see, when this blastula will enter, that means it will get completely embedded. So the trophoblast is going to give out these finger-like projections. What is this? These finger-like projections are known as chorionic villi. Is that clear? Finger-like villi means finger-like projections. These are known as chorionic villi. These are formed from trophoblast. And just like trophoblast was meant for providing nutrition, chorionic villi will also get into the uterine wall like this. It will interdigitate like this, right? So these are the chorionic villi. They are going into the uterine wall, tightly fitting into the uterine wall to draw nourishment from the mother's body, right? And this is chorionic villi. This will later on forms placenta along with other membranes, right? It also secretes HCG. This HCG will give you the pregnancy test, right? So what is the function of HCG, guys, here? So chorionic villi secretes HCG, that is human chorionic gonadotropin. This HCG is going to act on corpus luteum. So this maintains corpus luteum, so that corpus luteum, which is present within the ovary, still corpus luteum, right? So that corpus luteum does not degenerate and keeps on secreting or producing progesterone. Because we have read it in the previous lecture, guys, that endometrial lining is produced by estrogen, but it is maintained by progesterone. And we need this endometrial lining to be maintained now, because we have implanted that blastula, the embryo, here. Now, that is why we call it pregnancy maintaining hormone. If by chance corpus luteum degenerates or the level of progesterone falls after implantation, the endometrial lining will slough off. It will come out through bleeding. And along with that bleeding, along with that menstrual flow, will come the implanted embryo, thus resulting in abortion, right? So that is why we call it pregnancy maintaining hormone. Is that clear? So who maintains that? HCG. So why do we call it human chorionic gonadotropin? Because in humans, this chorion layer which has been formed, chorionic villi will secrete this hormone, and it will act on the gonad. Which gonad? Ovary. It will act on the ovary, because within the ovary we have corpus luteum. Is that clear? So what is the target tissue? That is corpus luteum. Where is it present? Within the ovary. Please tell me these points are clear to all. Yes, guys. Is that clear to all? Right? So this is the function of chorionic villi. Now this, if there is a hormone in our body, this hormone will be secreted in the urine, right? So this hormone will give us pregnancy test. Pregnancy test. It is a urine test, right? So presence of HCG in urine marks the positive test for pregnancy, and we call it Gravidex test. It is also known as Gravidex test. Please tell me these points are clear. Yes, guys? Are we clear? Right? Now, moving on to these extra embryonic membranes. Okay. Now, before this point, so see, we have zygote, So zygote, after zygote, 2 cells, 4 cells, 8 cells, 16 cells, and 8 to 16 cells was known as morula. And then from morula formed blastula. You must know the sequence. And this blastula had 16 to 32 cells, and so on. And then comes the next stage, which is called gastrula. So what is gastrula? When do we call a stage, an embryonic stage, as gastrula? Now see, we have seen in the previous diagram, see, so earlier there was this big blastocele. This was a big cavity, fluid-filled cavity called blastocele. Later on we saw that the blastocele has reduced and there is another cavity developed called amniotic cavity, right? So blastocele is reducing here. And now, During its reduction, there will be a stage when this blastocele is totally obliterated, that means totally disappeared. So when the blastocele has disappeared completely, disappears, right? And instead, there is formation of germ layers. Germ layers have been formed. What are the germ layers? Ectoderm, mesoderm, endoderm. We are triploblastic, right? And from these germ layers, organ formation has also started, organogenesis, right? So when these characters are present in an embryo, we call it gastrula. So the disappearance of blastocele starts the gastrula, the beginning of gastrulation. Once the blastocele is gone, we say now it is a gastrula, and during gastrulation, germ layers will be formed, and from germ layers, guys, organ formation will also occur. Is that clear? And then after gastrula, guys, now it will start looking like a human figure, and we call it as fetus. When the embryonic development completes, and that will be followed by childbirth. Is that clear? So while these all developments are going on, there are some layers formed around the human embryo. These layers are called extra embryonic membranes or fetal membranes. These are 4 in number: amnion, allantois, yolk sac, and chorion. Is that clear? Now see, during its development, guys, what are the functions of these extra embryonic membranes? They will protect the embryo from desiccation, means dehydration, mechanical shock, exchange of gases, absorption nutrients, and elimination of waste. Are we clear? Are we clear here? Please tell me. Are these points clear to all? So these are the various functions performed by all the 4 layers. Clear to all? Now the whole development, see, I told you pregnancy starts from the time of implantation till the development is complete, that means till birth. This is called gestation period. So what is gestation? It is a time period between implantation and birth. So from implantation to birth, this period is known as gestation period, or we simply call it pregnancy. Average is 266 days or 38 weeks from fertilization to the, yes, is this point clear? Right? Now, during this development, guys, there is a very important structure formed, right? We see, as soon as blastula gets into the uterine wall, endometrium starts going into the uterine wall, forming these interdigitations, which are known as chorionic villi, right? And gradually this chorionic villi is going to form placenta, I told you. This placenta is going to help in exchange between mother and fetus, exchange of nutrients, gases, waste, right? So there is exchange of these things also. So this is the first function of placenta. Second function, this placenta is also known as an endocrine. It is a temporary endocrine tissue. Why temporary? Only formed during pregnancy, and it secretes certain hormones. So temporary endocrine tissue, and it's going to produce certain hormones. So what are those hormones produced by placenta and what are their functions? Now see here. Okay, so the hormones produced are placental hormones. See, placental hormones. All right. So what are the placental hormones? Estrogen, of course, progesterone. Later, during the end of the pregnancy, it will also produce relaxin. Then other than this, it's going to produce HCG, which we have already discussed, chorionic villi, and followed by placenta producing HCG. Then HPL, that is human prolactogen, right? HSG, somatotropic hormone. And so these are the hormones produced by placenta. Is that clear? Now during pregnancy, guys, during pregnancy, there are certain other hormones also which increase. So during pregnancy, cortisol, thyroxine, that will also increase. Thyroxine, cortisol, that is the stress hormone, they also increase. Are these points clear to all? These are important lines given in NCERT. And now what events occur during pregnancy? See, we are talking about pregnancy. We are saying it is the average duration is 266 days or 38 weeks, or in short, we can call it 9 months, right? So these 9 months have been divided into, guys, 3 trimesters. So 9 months of gestation can be divided into 3 phases. First trimester, second trimester, and third trimester. So first, second, and third month, then fourth, fifth, and sixth month, and then seventh, eighth, and ninth month. Now, for the normal development, certain things or certain events should occur at specific times, right? So what are the things, what are the events that are going to occur at different time periods? Let's see. For example, during the first month, guys, heart should be formed. So if these events are occurring at the prescribed time, so that means the baby development is absolutely normal. So first thing, heart should be formed when? In the first month. In the second month, guys, limbs, digits means fingers, should be formed. In the third month, guys, major organ systems should be formed. For example, limbs, external genitalia, these should be formed properly, and external genitalia are well developed. So by the end of 12 weeks, that is first trimester, these things should have been formed. Are these clear? Now, during the second trimester, see here, the first movement of the fetus takes place. Now this particularly happens during the 5th month. This happens during the 5th month, and this has been asked many a times. This is a PYQ. Then along with this, appearance of hair on the head, and usually observe hair on the head and first movements during the 5th month. Now at the end of 24 weeks, that is second trimester, body is covered with fine hair, eyelids separate, and the baby starts opening eyes, and eyelashes are formed. These things happen by the end of 24 weeks or second trimester, that means 6th month. These are the events of 6th month. Are we clear? And then for the third trimester, see, by the end of 9 months, here, so third trimester, fetus is fully developed and ready for delivery. Are we clear? So these are the events occurring during the gestation period. Now when the fetus is fully developed, it will stimulate a mechanism that is known as parturition. It will stimulate this mechanism of childbirth. So childbirth begins with the labor and series of strong rhythmic contractions. This whole process is known as childbirth or parturition. Now remember, this parturition is going to be a neuroendocrine mechanism, and the stimulus stimulus for childbirth comes from the fully developed fetus. Fully developed fetus. So who gives that signal? The first stimulus or the first signal for parturition comes from fully developed fetus. So how this thing is going to happen? See, it is induced. It is a complex neuroendocrine mechanism. Why? There will be neural involvement, that is nervous system is also involved, sending signals, and then there will be certain hormones also playing an important role. So signals, guys, let's see here. Signals for the parturition originates from the fully developed fetus, and second from the placenta. Number one and number two. There are two structures from where we get the signal, right? And this is going to induce certain mild contractions and known as fetal ejection reflex. So what happens during the fetal ejection reflex? So as soon as the message reaches your maternal pituitary, so maternal pituitary is going to release oxytocin, mother's pituitary is going to release oxytocin, and this oxytocin, guys, this oxytocin is going to stimulate uterine contractions. Uterine contractions. Can anyone name the layer of uterine wall which is under the effect of oxytocin? Can you name that? We have already discussed that yesterday. Yes, guys. Which is the layer of uterus which is under the effect of oxytocin? Very good, Maria. So that is myometrium. So myometrium will start showing contractions. Now remember, the contractions and maternal pituitary, so they will have positive feedback mechanism. So once it contracts, oxytocin is secreted, it will show contractions. Contractions will have positive effect, more oxytocin, more contractions, more contractions, even more oxytocin. So there is a positive feedback mechanism in between the two. Are we clear till here? Right? So eventually, see, these are the steps followed: fully developed fetus and placenta. So these are the two signal generating structures resulting in fetal ejection reflex. So posterior pituitary releases it, not produces, it releases oxytocin, and oxytocin is going to affect the myometrium, causing strong uterine contractions resulting in expulsion of the baby through birth canal. Please tell me these points are clear to all. And as soon as the birth happens, the baby has to be fed by the mother's milk. So milk produced during the initial few days, guys, of lactation, that milk is known as colostrum. It has several antibodies, and which particular antibody? IgA, because it is a secretory antibody to protect the baby from initial source of infections. So that means the first type of immunity which we are born with, or not born with, which develops in our body is the passive immunity. Yes, the first type of immunity that develops, so that is passive immunity. Active immunity is going to develop gradually when we'll be exposed to the pathogen through clinical exposure, and even before that, vaccines, right? But mother is going to provide us with passive immunity. Is that clear? So breastfeeding during the initial period is recommended by doctors for bringing up a healthy baby. Is that clear? So all these lines should be well read and remembered, guys, because we have seen the pattern of the paper. NCERT is giving, NTA is giving direct lines from NCERT. So the line might look very common or very simple, but you have to know the NCERT lines very clearly. Is that clear? So don't ignore any line. Don't read your NCERT like a newspaper or just like a novel reading. No, you have to stop at every sentence, read it carefully, remember what an NCERT is saying, read in between the lines, and remember the line as such as well, so that you can quickly mark the answer. Are these points clear to all? So guys, these are very important recommendations for clearing our NEET exam, right? All right, guys. So with this, we complete the chapter of human reproduction. And now moving on to the next chapter, that is reproductive health, which totally depends on the first chapter, the concepts that we have developed in the first chapter. Are we clear here? Please tell me, are we clear here? Yes? John Devon, we will be completing all the chapters, but we have good amount of time, so we can revise things in depth. See, these are revisions, but in these revisions, guys, we are discussing all the chapters in detail. So we don't have to rush through the chapters. The quick revision will come later on. As we approach towards the exam, quick revisions will be done. We have so many things coming your way. There are so many things coming your way. Just stay tuned, subscribe, like, you know the drill, right? But the point is stay, trust, and keep learning. There is a proper plan for bouncing back in the re-NEET, whatever our score was, whether we were at 300 or we were at 500 or we were already scoring 700, but now we are going to give it back to NTA. We are going to get better, right? So if you are at 700, you have to score 720 out of 720 this time. Is that clear? So coming back here, so in this chapter, guys, first of all, let's understand what is reproductive health. And as per the definition of WHO, it is a total state of total well-being in all aspects of reproduction. What are the aspects? Physical. So you must know all the four aspects: physical, emotional, behavioral, and social. So now this statement might seem very simple, right? But when we are sitting for the exam, when we are taking the exam, these four words can also take away our mental peace, right? And that time we will not be able to recollect what were those four words. So remember, it is physical, emotional, then behavioral, and social. Is that clear? So you have to learn these as well. So what is it? It is PEBS, PEBS. Is that clear? Physical, emotional, behavioral, and social. You know, this is the trick of the exam. Even the simplest thing can really make us fall and get anxious, because while we are reading, we'll just quickly skim through it. We'll not pay attention what are the four factors or the four aspects. So what are the four aspects? PEBS, PEBS, right? Now moving on to the next. Yes guys, so India was the first country to initiate reproductive health program, which is known as family planning, and in which year? 1951. Later on, guys, it was renamed to be RCH, that is Reproductive Child Health Care. So this was renamed as RCH. Is it clear? So what is the objective and what are the things that we are going to take care in this? So such programs deal with awareness about reproduction related aspects for creating reproductively healthy society. Second, about birth control, care of the pregnant mother, postnatal care, importance of breastfeeding and equal opportunities for male and female child, and creating awareness of the problems due to population explosion, social evils, sex abuse, and sex related crimes. Are these points clear? So now this chapter has lot of ratification based things also, so pay more attention. Now then, so see, aims and needs of the sex education in schools. Why do we need it? Because the right information will help us to avoid the sex related myths and misconceptions, proper information about the reproductive organs, adolescence and related changes, and safe and hygienic sexual practices, STDs and all this. That is why we need sex education in school. Now there is some data also given here that in 1900, guys, in 1900, the world population was around 2 billion, that is 2000 million, right? And in 2000 it became 6 billion. So from 2 billion it became 6 billion. Is that clear? So you have to learn this, write them separately. Then in India, that was about world. In India, guys, population was around 350 million at the time of independence, that is, let's say 1947. Around this time it was 3 million, and by 2000 it has reached 1 million. So from 350 it has reached 1 billion, and by 2011 it has reached 1.2 billion. So that means, now this is the NCERT line, every sixth person in the world is an Indian. So that's how prominent we are in the world, right? So are these points clear? So are we noting down these lines carefully, right? So according to 2011 census report, population growth rate was less than 2%, right? And the rate of which population could increase rapidly. Is that clear? Then what are the reasons for population explosion? Now very important, increased health facilities and better living See, increased health facilities and better living conditions. Second, decrease in death rate because of health facilities and living rate. Death rate has declined. Maternal mortality rate has also declined, and infant mortality rate has also declined. So what is maternal mortality rate? So mother dying during the childbirth or within first month of child delivery, that is maternal mortality rate. And infant mortality rate is also the baby dying within the first period of its birth, first year of its birth, that is infant mortality rate. Now why has these two decreased? MMR has decreased and IMR has decreased and overall death rate has decreased because of better health facilities, more awareness and better living conditions, and increase in the number of people in the reproducible age. So more the number of people in reproductive age, more will be the population. So these are some reasons of population explosion. Now what are the control measures? Motivation. Motivate them to have smaller families and use the contraceptive methods. Awareness through slogans like hum do hamare do, we too are two. Then couples should adopt one child norm. Well, this norm has already been adopted in some countries. And then raising the marriageable age. So like 18 years and 21 years for female and males respectively, but there are many considerations by government at different times to raise this. Yes. Next guys, next we have contraceptive methods, and now this is a very important part of the chapter. Listen. So what are contraceptive methods? Contraceptive methods are called birth control methods. These are birth control methods. See, for what are the properties of birth control methods? These birth control methods are going to prevent pregnancy. This is not after the pregnancy has happened, this is to prevent pregnancy. So to prevent pregnancy, an ideal contraceptive, how does an ideal contraceptive look like? It should be user friendly, easy to use, easily available in the market. It should be effective, that means it should prevent pregnancy. It should be reversible, that means once we discontinue the use of contraceptives, pregnancy should happen. Okay, then it should have no or least side effects. And then it should not interfere with the sexual drive, desire, and the sexual act. Are these points clear? So these are the properties of ideal contraceptive. Now, also, it should be cost effective. So not very costly, then it will discourage the people or the population from using it, right? It should be easily user friendly also, so that most of the times the user doesn't have to go to the doctor every time. Is that clear? So these are the properties of an ideal contraceptive. Are these points clear? Right? Now, if we go with these properties, then as a matter of fact, guys, IUDs are one of the most accepted methods. But condoms remain still the most used methods, right? Why? Because for IUDs also the user has to go to the hospital at least once to get it inserted in the uterus. But as far as condoms are concerned, they can be used in complete privacy. Is that clear? Now, moving on to contraceptive methods. So these are the different types of contraceptive methods that we are going to study. Natural or the traditional methods, then we have barriers, we have intrauterine, oral contraceptives, injections, and implants. In oral contraceptives, we'll also talk about emergency contraceptives. And then we have surgical methods. Is this point clear? We also have surgical methods. Now guys, please see, why do we call natural methods as natural methods? First of all, because there will be no external device used. No external device used. Is it clear? Talking about barriers, so barriers are going to act as a barrier between sperm and the ovum. So here we have basically two types of barriers, one being mechanical barriers and then chemical barriers. So we'll talk about those, right? And then oral contraceptives. Now, out of these methods, which ones are for males and which one are for females? So in natural, we have both for male and female. For barriers are again for male and female. Intrauterine only for females, only for females, only for females as of now, and again for males and for females. Now, the point is, how are these methods and what do these methods do basically, right? So for that, we'll have to go into the details of each and every method and see what is their mode of action. So first of all, starting with the natural or the traditional method. So why do we call it natural? As I told you, no external device used. And what is the mode of action? What does it do actually? What is the mode of action? So the mode of action is that all the methods are going to prevent the meeting of sperm, avoid the chances of meeting of ovum and sperm, that is, prevents fertilization. All of them will prevent fertilization. See, as we progress through the contraceptive methods, we'll see there are certain methods which will, like this one, it will prevent fertilization, while there are others which will not let the sperm enter into the female tract, right? There are others, even if the sperm enters, it will reduce the fertilizing capacity and suppress their motility so that sperm is not able to reach the ovum within the viability period, right? So, or there will be other methods which will prevent ovulation, which will increase the consistency of cervical mucus so much that sperm cannot swim through. So different methods are going to have the different modes. So what is this method going to do? It is going to prevent fertilization. How, why? We'll see it through. Now, so different methods, natural contraceptive methods are, first is periodic abstinence, second is coitus interruptus, and third is lactational amenorrhea. This is for female, this is for male, while this is also for female, right? Now, talking about the periodic abstinence, see here. So what does periodic abstinence mean? We have read menstrual cycle. We know ovulation occurs between 14 to 16 days. So when we know ovulation is occurring between 14 to 16 days, so that is the time when there is maximum chance of fertilization, right? So periodic abstinence means avoid coitus in the time period near ovulation. So avoid coitus between 10th to 17th days of menstrual cycle. So we call 10th to 17th day as, we can call it unsafe period, or we can call it fertile window, right? So avoid coitus during this period, simple as that, right? Why? Because ovulation occurs around this period, because ovulation occurs between 14th to 16th day. Please tell me these points are clear. Yes. Now this is according to the menstrual calendar of the female, hence we call this method as calendar method. Also we call it rhythm method because this is according to the menstrual rhythm of the female, rhythm method. So these are three names for the same thing. Talking about coitus interruptus, so here male has to withdrawal of penis just before ejaculation, so that insemination is prevented, right? So why? To prevent insemination in the vagina, so that semen or sperms are not released in the vagina, to prevent insemination in female vagina. Are we clear? Right? This is coitus interruptus. So it is also known as withdrawal method. Third is lactational amenorrhea. Amenorrhea means absence of menstruation. Absence of menstrual cycle is amenorrhea. When? During lactational period, that is during period of intense lactation. And what is the period of intense lactation? That is first 6 months after childbirth. Now why? Why is it so? Because, see, you must know the reason. So, all these are assertion reasons. This is assertion, this is reason. This is assertion, this is reason. This is assertion, this is reason. Can you see? We have 3 assertion reasons forming here, right? Now see, because during the period of intense lactation, prolactin is high. Prolactin high, and high prolactin, F will suppress FSH. So low FSH means no menstrual cycle. Please let me know all these points are clear to all. Yes guys, please tell me these points are clear. Yes or no? All clear? Only Maria is answering properly. What happens to other? What happened to others? Busy in chit chat, forming new friendships. Very good time for that. Anyways, so guys, these are natural traditional methods. Now we call them natural methods because there is no external device used. Now what is the advantage of this method? The advantage is no side effect. Since we are not using any external device, there are no side effects. So this is the advantage. What is the disadvantage? The disadvantage is high failure rate. High failure rate. Not very effective. Why? See, first of all, periodic abstinence is only effective. I'm writing down here. So the disadvantage of periodic abstinence is it is only effective in females with regular menstrual cycle. Only if a female has regular menstrual cycle. So this is the disadvantage. Coitus interruptus. So male has to withdraw penis before ejaculation. What if the male forgets to? So male may not be able to withdraw at the right time. And some ejaculation may occur in the female tract. What about lactational amenorrhea? So as per the theory, it is the first 6 months. No, it is hardly effective for first 2 months, or there are cases when pregnancy occurs right in the 2nd month of childbirth or 3rd month. So only effective for first 3 months. That is also theoretical only. So these are all the disadvantages. Please tell me these points are clear to all. Yes guys, is that clear to all? Right? All right. Now moving on to the next, which is, so we are going to talk about the second type of contraceptive method, which is, wait a second guys, it's really hot. All right, so now we are going to talk about the second method, which is called the barrier method. We are going to talk about the barrier method here. Right? All right. So barriers, these methods are going to prevent physical meeting of sperm and ovum. So basically, what are these also going to do? These will also, hashtag, they will also prevent fertilization. The same mode of action is exactly the same. Mode of action. Are these clear? What are the different methods included? Condoms for male. These are for female. While there can be female condoms also, which are known as femidomes. These are for females, right? If we talk about condoms here, so see, condoms, they're made up of rubber and latex. Male ones cover the penis. So this will collect the semen and prevent insemination in female reproductive tract. Prevent insemination in vagina. Is that clear? Then we have femidome, female condom, covers the vagina and cervix. They are just used just before coitus, prevents the entry of semen in the female reproductive tract, and particularly where? In vagina. So Nirod is a popular brand name for condoms for males. Is that clear? These are all NCERT lines. Now talking about why condoms are very popular, because it protects the user from STDs and AIDS. And guys, this is the only method, hashtag only method that prevents STDs. Along with preventing pregnancy, it prevents STDs. There is no other method that can serve the same purpose. Are we clear? Are we clear here? Right. Then they are disposable, that means used only once. They can be self-inserted and thereby it gives privacy to the user. Is that clear? So these are condoms for males, these are condoms for females. Then talking about other methods which are also used by females, these are all for females, which is diaphragm, cervical caps and vaults, made up of rubber, okay, and rubber, and then inserted into the female reproductive tract to cover the cervix. Now see, they will block the entry of sperms through cervix. Hashtag sperms enter vagina. Enter vagina though, but not the uterus, because at cervix there is a blockage of these structures. Now these ones are reusable, and along with these structures, guys, all these mechanical structures, we can use these chemical barriers. These are chemical barriers, and chemical barriers are going to increase the contraceptive value of these structures. They are going to increase the contraceptive efficiency of mechanical barriers. They are not used in isolation. Hashtag not used in isolation. Means the person cannot use just cream and jelly and foam and think of contraception. No, it won't be effective in that manner. Are we clear? Then talking about the third, very important. This topic is so important that we usually get question from this topic every year. Now, intrauterine devices. So first of all, the point is intra means within. So device is placed, inserted by through vagina into the uterus. So where are they placed? As the name says, they are placed or inserted in uterus. Can they be used by the person himself? No, they are inserted by the doctors or nurses. Is that clear? In non-surgical manner. Then it increases the phagocytosis of the sperms, and ideal method to prevent pregnancy. Told you, one of the ideal methods and one of the most commonly used methods. So guys, now there are three different types of IUDs. One is non-medicated. So in non-medicated, we have Lippes loop in the name of the person. Here we have copper T, copper 7, multi-loads with different potencies. Is it clear? And in hormone releasing, we have LNG20 and we have Progesta-SERT. And we have Progesta-SERT. Are we clear? These are the names. The names should be well learned first. Now all these IUDs, they work in different ways. So non-medicated, or we also call them inert IUDs. These IUDs, they increase the phagocytosis of sperms. In uterus. So it makes the uterus more hostile. Makes uterus hostile towards sperms. Hostile means non-friendly. Is that clear? Talking about copper releasing, so basically these copper ions, what do? Copper ions, they suppress the sperm motility. suppress the sperm motility. So the sperm is not able to move quickly towards the oocyte, right? Suppresses the sperm motility and the fertilizing capacity of sperm. Is that clear? So both are suppressed. Then hormone releasing, what do they do? Hormone releasing are basically, number one, going to make uterus unsuitable for implantation. And second, it is going to alter the cervical mucus. So the cervical mucus will become so thick that the sperms won't be able to swim through them. Are these points clear? So you should clearly know what are the different modes of action of each IUD. Is that clear? Now coming on to the side effects first here. So talking about the side effects of IUDs. Yes. So guys, okay, side effects are here. So advantage is that, okay, they are effective of course, but the side effects are because we are placing these external devices in the uterus, there are cases in which there are perforations in the uterus, some ulceration takes place in the uterus, or it can lead to heavy menstrual flow, or due to heavy menstrual flow it can just get removed automatically, and it is so small that the female might not even notice the removal of IUD, and according to her IUD is present in the body but it's not. So it can result in unwanted pregnancy. So then we have these methods called oral contraceptives, also known as pills. So basically there are two types of pills here, two types of pills. One is steroidal and the other are non-steroidal. So the non-steroidal one is Saheli. Why do we call it Saheli? Because it is friendly, more friendly, less side effects. Actually the name is centroman. Okay, while there are some steroidal pills. So the steroidal pills basically contain estrogen and progesterone. Now in steroidal pills also we have two. We have mini pills which contain progesterone only. And we have combined pills containing progesterone and estrogen. And estrogen. Are these clear? So different pills have to be taken in different ways. For example, see, oral administration or the combination in the form of tablets. So they're taken daily for 21 days, starting within the first 5 days of menstrual cycle, and after 7 days should be repeated. Now this is a pattern for combined pills actually. Mini pills are taken every day. They are taken every day without any break. While if we talk about Saheli, guys, Saheli is taken on a weekly basis, once in a week pill. It is taken once in a week. Are we getting it? So different pills have different ways. Now, what is their mode of action? They inhibit ovulation and implantation, and they thicken the cervical mucus to prevent the entry of sperms. They are very effective with lesser side effects. Well, that is theoretical. Are we clear? Now talking about Saheli, as I've told you, it is a new contraceptive method developed by CDRI, Central Drug Research Institute, that is in Lucknow. It is non-steroidal, told you already, once in a week pill with very few side effects and high contraceptive value, and that is why the name is Saheli, because of very few side effects. Is it clear? So again, assertion and reason. Are we clear? Then the same hormones which we are taking in the form of pills, they can be taken in the form of injections, like once in a month or twice and so on. So the combination will remain the same. The injection will either contain only progesterone or it will have a combination of estrogen and progesterone. Is that clear? And if we don't want an injection, we can go for the implant. Now these implants are under the skin, so they are subcutaneous. And implants are basically considered more effective because since they are under the skin all the time, so they are continuously releasing some hormone, so the effectiveness is very high. So see, it is just like pills and their effective periods are much longer, and this is how we are taking them. So the same combinations. Is it clear? Now talking about emergency contraceptives. See here, emergency contraceptives. So what are emergency contraceptives? So these emergency contraceptives are basically post-coital pills. So we call them morning after pills. That means they are taken post-coitus, after mating. And when there are chances, okay, it might result in pregnancy, that is where the role of emergency contraceptive comes in. It should be taken within the 72 hours of coitus, but the sooner you take, more is the effectiveness. So it avoids pregnancy due to rape or casual intercourse. Now they also contain the same hormones but in very high dosage. Is it clear? They're taken in very high dosage. For example, I pill, E pill, unwanted 72. So why do they put this figure 72 here? It is because of their effectiveness within 72 hours. Also guys, IUD, if placed after coitus or within coitus, sorry, placed within uterus within 72 hours of coitus, can also act as emergency contraceptive, can also be an emergency contraceptive. Are we clear? Is that clear? So not only these pills, even IUDs can act as an emergency contraceptive if placed within 72 hours. Please tell me all these points are clear. Yes guys. Yes, Aswant, LNG20 means levonorgestrel, and levonorgestrel is a name for synthetic progesterone. Is that clear? Now, so guys, these are surgical methods. Let's talk more about it. So surgical methods, guys, sterilization. These are called permanent methods. Why do we call them permanent methods? Because the reversibility is very poor. Again, assertion reason, because of poor reversibility. Because of poor reversibility. What do? They block the gamete transport. Therefore, that is, they are also hashtag, they also prevent fertilization. So there are three methods that we have discussed which work on the same mode of action. All the three methods which prevent the fertilization. Number one, natural methods. Number two, barrier methods. And number three, surgical methods. All three of them has this one thing in common, that they will prevent fertilization, right? Very effective, but reversibility is poor. Hence, we call it permanent. Why do we call it surgical? Because it does involve a minor surgery. So for males, guys, we have vasectomy, and for females, we have tubectomy. In vasectomy, what will happen? Vas deferens is cut and tied. Cut and tied. While in tubectomy, it is the fallopian tube that is cut and tied. Are these points clear? Vasectomy and tubectomy. So this is the picture given in NCERT. Vas deferens, cut and tied, right? So this is the method in males, right? And then if we look at tubectomy, here fallopian tube is cut and tied like this, right? Now the points to be taken care of here: after vasectomy, semen is produced. So these are statement based questions about vasectomy. So semen produced even after vasectomy, but semen doesn't contain sperm. Semen does not contain sperms, right? Similarly, after tubectomy, menstrual cycle continues. Normally, very normally, there will be no difference. After tubectomy, menstrual cycle continues, but menstrual flow will not contain, menstrual flow does not contain the unfertilized secondary oocyte. Are these points clear? So that is about tubectomy. Clear? So what are the different, guys? So these were the contraceptive methods. What are some of the side effects of these contraceptive methods? Nausea, abdominal pain, breakthrough bleeding, that is in between. Random bleeding might occur other than your menstrual bleeding, and irregular menstrual bleeding, and it can cause breast cancer because of high doses of estrogen in the pills or injections. Is this clear? And then we have MTP. Now MTP is not a contraceptive method. It is a method which is going to which is used after pregnancy has occurred. Okay, if unwanted pregnancy has occurred, then we can go for this method, that is MTP. It is the intentional or voluntary termination of pregnancy before the full term, that is MTP, also known as induced abortion. So these are some NCERT lines that you should remember, that 45 to 50 million MTPs are performed in a year in the whole world, all over the world. That is 1/5 of the total number of pregnancies. So it helps to decrease the population, and it is not legalized. Many countries do not legalize MTP due to emotional, ethical and religious and social issues, but Government of India has legalized it, understanding that population won't stop and rather use some illegal and non-medical ways which can be at the cost of the mother. So Government of India has legalized it in 1971 to check the illegal female feticides. Is that clear? Now see, what is MTP? To avoid unwanted pregnancies due to casual intercourse or failure of the contraceptive, and essential in cases when continuation can be harmful to the mother or to the fetus or both. Is it clear? Now problems related. What are the problems related? So majority are performed illegally, and then misuse of amniocentesis test for the fetal sex determination. So such practices are dangerous for the young mother and the fetus. Is it clear? Now what is this amniocentesis? So amniocentesis is basically a prenatal diagnostic test. It is a prenatal diagnosis for genetic disorders. Is it clear? For the genetic disorders like sickle cell anemia, hemophilia, right, thalassemia, or Down syndrome, Klinefelter syndrome, all those are checked through these amniocentesis. So what do we do in this method actually? So here amniotic fluid of the fetus is taken out to analyze the fetal cells and used to test the genetic disorders like and the survivability of the fetus. Now these are important points. Government of India has enacted the MTP Act of 2017. What does it say? To reduce the illegal abortion and consequent maternal mortality and morbidity. In that case, according to this act, so if MTP is being performed within, see, if it has to be terminated within the first 12 weeks, that is the first 3 months, in that case, only opinion of one registered medical practitioner is fine. So if you are going for abortion in the first 3 months, and the first 3 months are considered rather relatively safe for MTP, so we need opinion of one doctor. But if it is between 12 to 24 weeks, that is in the second trimester, then it has to be 2 registered medical practitioners giving that opinion. Is that clear? So these are the functions, sorry, these are the rulings of Act 2017. Is it clear? Then there are some STDs, guys. So what are STDs? STDs are also known as sexually transmitted diseases, or we call them as sexually transmitted infections, or venereal diseases, also known as RTIs. So what are the examples? Gonorrhea, syphilis, genital herpes, chlamydiasis, warts, trichomoniasis, hepatitis B, and HIV. You must know that the age group where STDs are found maximum is between 15 to 24 years of age. This is the age group which is most vulnerable for STDs. Is that clear? So how do these spread? So guys, by the way, they spread through sharing of injections and needles and surgical instruments, transfusion of blood from mother to the fetus, and there are three STDs which are totally incurable, which are, so how do we remember incurable STDs? I remember it as 3H. How do we remember the three STDs which are incurable? 3H. One is herpes, second is hepatitis B, and third is HIV/AIDS. So these 3 STDs are completely incurable. As far as other STDs are concerned, if they are diagnosed at the right time, we still have their good treatment. But these, whenever you diagnose them, they are incurable, non-curable. Are we clear? So you have to remember 3H. That is herpes, hepatitis B, and HIV/AIDS. Clear? All right. Now, moving on to the symptoms. So what are the symptoms here? So the symptoms are itching, fluid discharge, slight pain, swelling in the genital region, and so why do these get incurred or not cured properly? Because of the social stigma attached and absence and less significant early symptoms. So people ignore these symptoms, and even when they find out the symptoms, so there is a social stigma attached to it. What if people get to know? What will others think? So they don't get that treatment done. So what are the severe complications related to it, guys? One is pelvic inflammatory disease, PID. It can also lead to abortions. Stillbirth means dead baby is born. Ectopic pregnancy other than in uterus. Infertility, cancer like cervical cancer. And how to prevent, we also know. Avoid sex with unknown partners. Use of condoms should be practiced. And then go to the qualified doctor for early detection and get complete treatment done. Is that clear? And then talking about infertility, it is the inability to conceive and produce children even after 2 years of unprotected sexual cohabitation. Right? The reasons could be physical, congenital, that means by birth, drugs, immunological, or psychological reasons, and it can be treated by the techniques known as assisted reproductive technology called ART. Now, let's talk about ART in detail. Very important topic here. So guys, ART consists of 2 types of techniques. We have in vitro fertilization, and we have in vivo fertilization. So in vitro means here fertilization will occur in lab conditions. That means outside the female body. And in vivo means in natural conditions. That is within the female body. Is that clear? Now, so both can be simply referred to as IVF, in vitro or in vivo. Now, first of all, you should know that which methods comes under which category. In vivo, guys, we have test tube baby program, and second is ICSI. Is that clear? Now both these methods are followed by, embryo transfer. Followed by embryo transfer, and this embryo transfer can occur by ZIFT and IUT. These are the two methods of embryo transfer. While in this we have GIFT, AI and IUI. So please be careful, IUT and IUI, they are two different methods, and what does each method involve? Let's talk about this. So in test tube baby program, guys, so basically we are going to collect egg from the female donor or the wife, and similarly egg from the sperms from the male or the male donor, and then this is all done by hormone therapy. So female is given lots of hormones, FSH, and multiple ovulation is induced before the retrieval of the egg, right? And then both eggs and sperms are made to fertilize in the test tube. So basically, why do we call it test tube baby program? Fertilization in test tube occurs in test tube. So there will be multiple embryos formed. See, multiple embryos are formed, and after we check the development of these embryos, we are going to transfer this embryo. This will be embryo transfer. We will transfer this embryo back into the female body. Is it clear? So this is in vitro fertilization, and which one? Test tube baby program. Now this embryo transfer can be of two types. Now if we transfer zygote or the 8 blastomere stage, so normally zygote and 8 blastomere stage is present in fallopian tube, right? So if we place this stage back into the body, so where are we going to put it in? In the fallopian tube. So zygote or 8 blastomere stage is placed back in fallopian tube. That can be one. Or we can still carry on with the embryonic development outside the body and wait for a bigger embryonic stage. So if we transfer more than 8 blastomere stage, then that will be placed in the uterus, right? 8 blastomere stage is placed in the uterus. So depending upon which stage of embryo is being transferred into the body, we have ZIFT and we have IUT. So ZIFT is zygote intrafallopian transfer. See, in this stage we are transferring zygote or 8 blastomere stage in the fallopian tube, and hence the name, right? And then we have IUT. In IUT we are transferring the more than 8 blastomere stage in the uterus. So this is known as intrauterine transfer. Are we clear? Are we clear? So these are the two types of embryo transfer. Is it clear? Then another method is, so this is what we have already seen. Then second method is intracytoplasmic sperm injection. Now in this method we are going to take just one sperm in the microneedle, and we are going to put it into the cytoplasm of sperm under microscope. So do we see, this is that needle, and through this needle we are bringing one sperm into the cytoplasm of egg, right? So intracytoplasmic sperm injection, the name itself says sperm is directly injected into the cytoplasm, injected directly into the cytoplasm of ovum. Again, fertilization is occurring outside the body, so it is in vitro. Is it clear? Now, zygote is formed and it will be again followed by embryo transfer. You can either use zipped method or you can use IUT. Are these points clear? Then guys, next method that we have is gamete intrafallopian transfer. So what happens in gamete? Now in gamete intrafallopian transfer, do you see a catheter here? So with the help of catheter, we are going to place the egg. We are going to place one egg. This is the egg from the female donor. So from the female donor, we have taken an egg and we are placing it in the fallopian tube of a female who cannot produce one, of a female who cannot produce ova. So there is a female who has absolutely normal body to carry out the pregnancy, but she is not able to produce egg. So what do? We collect the egg from a female donor. With the help of a catheter, we are placing it in the fallopian tube. This is called gamete intrafallopian transfer. So which gamete, that is egg, is placed intrafallopian, that means is placed within the fallopian tube, non-surgically, right? And then we send the female back home, normal mating will take place and fertilization will take place here. So because fertilization is going to occur in the female body, hence it is a in vivo method. Is it clear? So you should know which method will be employed when. So case study. Then artificial insemination. This method can be used when the male is having low sperm count or suffering from erectile dysfunction, right, ED. So in that case, artificial insemination can be a good deal. So basically, non-surgically, we are inseminating the sperms into the vagina. So in artificial insemination, sperms are placed in vagina. This is artificial insemination. Now better than this is if sperms are artificially placed deep into the uterus. Now it has better success rate because sperms are carried forward into the uterus, so they will not have to face this challenge of cervical mucus and all, right? They're placed into the uterus. Now this is known as IUI, intrauterine insemination. So of course this has a better success rate. So this is done when there is a low sperm count or the person suffering from erectile dysfunction. Is that clear? And then apart from all these methods, we have another method called surrogacy when we are using a third female. So here there is a third individual involved. So another female, that is surrogate mother, carries out the pregnancy, is going to carries out the pregnancy. So this surrogate female it's not her egg, okay? She's not the owner of that egg. Now this is the female whose egg has been taken, and this is the male whose sperm has been taken. They were fused outside, maybe let's say through test tube or ICSI. So in vitro fertilization, and then embryo was transferred. Embryo transfer was done in surrogate female. So basically surrogate female is lending her uterus. But the point is, this female should be healthy, because through placenta the diseases can travel. Is this clear? So this is the surrogate female. Are we clear? Please let me know these points are clear to you all. So guys, with this we complete the chapter of human reproduction and reproductive health, and we will be taking another class tomorrow, and in tomorrow's class guys we are going to discuss health and diseases and microbes and evolution probably. Is that clear? So we'll be taking a longer class tomorrow and discussing these 3 chapters, and then we'll be left with biotechnology. Is that clear? So thank you so much for joining me joining me today. And guys, another reminder, so now all India mock tests have been extended, that is thrice a week. That will be Wednesday, Friday and Sunday. Is that clear? So make sure that you are taking these tests on the decided days, and gradually as we proceed further, as we move towards a Re-NEET exam, all India mock tests will be happening every day. So get ready, give more tests so that you know your mistakes, you know what to really take out of these lectures. Is that clear? So you get a rank predictor, you get a proper video solution, you get a proper analysis, which is a very important thing for your exam, because this is Re-NEET and we have to bounce back. See you tomorrow at the same time, and goodbye.